Hi, I'm Peter Kharchenko. I'm a principal investigator at Altos Labs, San Diego Institute. We have a number of directions that we've been studying. I think for the most part, my group develops computational methods, but we try to develop it in the context of specific biological studies. So much of the focus for the past 5 years has been on analysis of cancer, looking at both metastatic solid tumors as well as hematological malignancies. And we try to use the single-cell methods to understand the sort of compositional effects, sort of the repertoire of cell types and states that exist in these tumors, and how they develop, how they react to treatment, and how they related to disease progression in general. And so in that context, we've developed methods for, let's say, comparison of sort of clinically oriented panels. This is what I've talked about here, as well as methods for analysis of genomic aberrations that I mentioned, tools for analysis of just inter-individual variation to sort of understand maybe the factors that distinguish patients and also factors that distinguish us within just normal population. So these are generic tools that can be used in all sorts of contexts, but they stem from our work mostly on these cancer cases. Currently, when people say multiomics in the field, what I actually mean is a very specific assay from 10x Genomics that measures chromatin accessibility and RNA sequencing. Now, of course, the term multiomics is much broader than this. So we do use multiomics in a broad sense, I think, looking at, let's say, protein and expression, and also, I guess, at accessibility and expression, looking at metabolites and expression, DNA methylation, and so on. But yeah, these data types are quite rare, so we're certainly not at the forefront of that type of analysis. I think the only real contribution that we hope to make is on this kind of vertical integration, which is called. So this is a question of how to take into account multiple molecular modalities measured for the same cell. So if you, let's say, measure expression in that cell, but also chromatin accessibility, and then you compare two different cells. So the question is how similar they are, and they have some similarity on expression state, they have some similarity on epigenetic state. How do you balance the two? And that is an important question, I think, and even just the philosophical interpretation of it is not very clear. So we try to we hope to come up with sort of benchmarks that are based more on this kind of rationale as to what we would want from this type of integration, and see how different integration methods actually measure up relative to that. Technologies. There are quite a few of these multi-omic assays that people have developed over the past few years, including this 10x commercial kit for accessibility. We haven't been developing those. I think we'd like to. There's a lot of room for improvement and so the new horizons that could be explored with respect to, let's say, states of not just the genome but also the RNA molecules. But yeah, those assays are difficult to develop, so we don't have them. The other big sort of area that is related to this are spatially resolved measurements. So those are mostly transcriptomic for now, but we're hoping to layer in some of the other modalities there as well. But again, it's very hard work, so I don't know if we'll succeed. Most likely somebody else will succeed, but that's great because we'll just use that. Yeah, so human brain is the most complex organ that we have, that we're studying. And the key challenge really, I think, is just managing this complexity. So my lab was also part of the ICCN Brain Initiative Cell Census Network. So Brain Initiative is a big NIH project for basically profiling all sorts of things about the brain, and the ICCN in particular is trying to create atlases, molecular atlases of the brain, as well as connectivity atlases. And so we had a little bit of contribution into this, and just the number of subtypes, let's say, of neurons, number of states there, it's quite quite difficult to handle. A lot of variation that we see within individual cell types, likely functional, but also related to spatial organization of the brain. The other challenge is that, especially with the single cell techniques that we focused on, really what we're seeing are just the sort of parts of neurons. All these processes that neurons have. We don't capture them. They have their own states. Close, let's say, to synapse or in the axon somewhere. There are different composition of, let's say, mRNA molecules that can be there, and we simply don't see this. Or if we do see their contribution, we cannot connect it to the cell body that we mostly analyze. So that's sort of, very partial view, and I think going forward will be a major challenge to connect that. So multiomics in general tries to get a more comprehensive measurement of cell state, and there are obviously a lot of things, in brain in particular, we don't understand about the state of these cells. So right now it's a basic science tool that can give us sort of classifications that eventually I think will need to be connected to function of neurons. That's really a major effort in the field, and once we have better understanding of how different subtypes of neurons relate to different function, even on sort of molecular electrophysiological level, that can potentially give us a much better way of looking at disease. And obviously the brain disorders are difficult to understand, both behavioral and neurodegenerative disorders, and understanding how different subtypes, how different elements of the brain is supposed to behave, at least in the healthy brain. That's going to be very valuable for looking at these diseases and seeing what's wrong. Do you have let's say a skew in terms of which cells are active or inactive, or do you have other cells invading somehow, proliferating where they shouldn't, or lack of proliferation in case where, let's say, myelination and so on. So all these, I think, molecular atlases that are being created by Multiomics will be helpful in trying to understand at least cellular function. Societal impact, that's much harder to predict. I mean, if this helps to make progress on treating any of these disorders, that potentially can alleviate large societal burdens, particularly with neurodegenerative disorders that are increasingly concerning. So hopefully it will give us some insights into this.